Opiates are analgesic alkaloids derived from Papaver somniferum. These have been used medicinally for thousands of years and are still in use today. They have a global market value estimated at $23 billion US. In this video, we will talk about the production of opiates synthetically in E. coli. First, we will discuss how opiates reduce pain, then look at how opiates are synthetically produced, and, by the example of salutaridine synthase, how this was done in E. coli. During pain, action potentials are transmitted through nerves. At the tip of each nerve, calcium channels open, leading to influx of calcium and release of neurotransmitters, continuing the signal. Opiates, however, bind to opiate receptors on these neurons, resulting in the closing of the calcium channels. The action potential is therefore stopped and no pain is felt. The pathway that produces opiates is as follows. Escochlorine is first methylated, then hydrated, and finally methylated again, resulting predominantly in S-reticulene. S-reticulene is then oxidized to dihydroreticulene and reduced back to R-reticulene. R and S-reticulene are stereoisomers. Salutaridine synthase then creates a phenol carbon-carbon bond, producing salutaridine. Reduction of salutaridine produces salutaridinol, which is then acetylated, and spontaneously converts to the opiate thebaine. Thebaine is a commercially significant opiate with an important market value. Here is where we stop. The role of salutaridine synthase is the conversion of R-reticulene into salutaridine. Salutaridine synthase is a cytochrome P450 and as such contains an iron heme group. The hydroxyl at the 1' position is attacked first of all by iron 4+ from the heme group. Immediately afterwards, the hydroxyl at position 7 is further oxidized by iron 3+ from the heme group. Cytochrome P450s such as salutaridine synthase oxidize through molecular oxygen, but also require the transfer of electrons from NADPH in order to form the highly reactive iron 4. Expression of proteins in prokaryotes such as E. coli can be very difficult. First, codons are present in different abundance in different organisms. As such, the DNA extracted from the poppy plant might contain codons that are extremely rare in E. coli. Therefore, translation is slow since it cannot find these rare codons. The DNA transcript was therefore optimized to use highly abundant codons in E. coli, optimizing translation. In addition, cytochrome P450s such as salutaridine synthase are bound to the endoplasmic reticulum, and E. coli do not possess an endoplasmic reticulum. In order to produce functional protein in E. coli, the N-terminal domain that corresponds to the ER transmembrane domain must be cleaved off. This is achieved by PCR. A primer that binds after the N-terminal domain does not replicate the N-terminal domain section of the gene. The primers used also include a modified 5' region that adds a start codon to the DNA, allowing translation initiation. In order to transform E. coli to express salutaridine synthase DNA is inserted into a plasmid containing an ampicillin resistance gene. Heat shock then allows uptake of the plasmid by facilitating its movement through membrane pores. Transformed E. coli is grown on a medium containing ampicillin, which selects colonies that have taken up the plasmid. Salutaridine synthase expression is induced by the addition of IPTG. Salutaridine synthase is designed to be expressed in tandem with salutaridine reductase and salutaridine acetyltransferase, resulting in an E. coli strain that produces thebaine from R-reticulene. R-reticulene can be produced by transformed E. coli strains from tyrosine. Therefore, the complete production of opiates is possible in E. coli.